T-minus 30 seconds. T-minus 20 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0.
You are now tapped into the coolest reptile podcast in the world. Welcome to the Trap Talk Reptile Podcast's Monday nights new breeder on the block series, only more like new genetic on the block series, if you know what I mean. This is going to be an epic episode, guys. This is episode 353. Hope you guys are ready. I'm your boy, MJ. What is good? If this is your first time tapping in, do me a favor. If you're into keeping reptiles, breeding, admiring, overall just keeping, all the above, then you're going to want to hit that subscribe button, smash that notification bell. Let's get the likes up right now. Let's get that over with. Free feature. Boom, likes up right now. And then drop a comment. Let me know what you like best. Let me know what you're getting out of every single episode. But it's all about the growth, all about evolving, man. If you're in the reptile industry, then you should be tapped into this podcast straight up. So I just want to say thank you for all the love and support. Shout out to all my subscribers out there. Shout out to everyone in the early birds. I know you guys are ready to rock and roll. But before we get into things tonight, I will say that tonight's episode is brought to you by my boy, Miguel Garcia. Always evolving pythons, always doing heavy next level things. It's really to see Miguel really take things where they are at right now for him. He came in probably maybe a year or so before I did. And, you know, I've always looked up to him since I've been in the game. And he's really just has lived up to the expectations as far as, you know, being one of the leaders in the industry for sure, man. He's in Mexico, been in Mexico, back and forth, doing big things over there. And it's just very inspiring, I have to say. So, Miguel, thank you so much. Tina, the entire AEP family. I appreciate you guys for always being loyal to the trap and supporters of mine. And man, I look at you guys as family. Thank you so much. I also want to say that number one stainless steel RHACS in the game is Freedom Breeder, the original Freedom Breeder since the 90s. Okay, if you got a Freedom Breeder rack in your collection, you're doing big things. Okay, next level of reptile keeping or breeding rodents. Okay, I got to get my rodent rack from them. I'm not gonna lie, I'm pretty stoked. But anyways, guys, this is it. The official rack company of the industry, Freedom Breeder. Number one, pat yourself on the back if you got yourself a Freedom Breeder rack. And I will also say that I'm excited to say that I put my 6 clutch of ball python eggs in my sim container. That's right. Shout out to sim container. If it's a sim, it's a win. Less steps, less stress. If you're winning, you're simming. Did I say that right? I don't know. Listen, John, Alex, I appreciate you guys so much. Next level monitor game happening at Sim Container or Sims. Go to Instagram, go follow them, and just amazing stuff over at Sims. They've been in the game for years, so mad respect to them. If you're looking for exclusive content, if you like to support what I have going on, if you want to tap in with some really hardcore, dedicated animal keepers, lovers, all the above, then do me a favor, do yourself a favor, especially if you want to tap in with some and kind of get your name out there more, dude. Nothing will top joining the Trap Talk Patreon family. I got OGs, little OGs, and soon to be OGs, all a part of the Trap Talk Patreon family. Either way, we all want each other to grow. Want each other to grow. Biggest thing about joining the Trap Talk Patreon family is once you join and you get that link, you get the link to the Discord, and the Discord will tap you in with over 165, over 170 now, over 170 trappers a part of the Discord, multiple channels, nonstop reptile talk going on. It's so much just to get out of as far as if you wanting to be a part of this industry. So, trappers, my family, love you guys. Thank you for all your love and support. Who's ready for the Reptile Super Show? I'm trying to tap in with all my trappers at the Reptile Super Show. Number one show in the country, baby. Let's go. Cali, where you at? Happening Anaheim, July 8th and 9th. Okay, I'm going to be vending. That's right. I'm going to be vending. I'm going to be posted up right there next to my boy, Matthew Summers. He's going to have some heat for sure. I'm going to have some heat, and I have a special guest who's going to have some heat. Just saying. I don't want to, if he wants to say it, then he could say it. But I ain't saying shit. Just be ready, okay? I'm pretty happy to have someone pretty legendary be standing next to me at my table in Anaheim. And you got to go there to see who it is, okay? But either way, it's going to get cracking. Go follow me on Instagram, TrapTalkGod619, to stay on top of all my projects. Follow the podcast's Instagram page as well. Trap Talk Reptile Podcast. Facebook, what is up? If you're on Facebook, hit me with the follow, hit me with the like, and I appreciate all the love and support coming from Facebook. Let's go. Early birds, who's here? Listen, if you're in the live chats tonight, let's get those super chats up, right? If you have an important topic to bring to tonight's table, if you think you could bring some heat that we could talk about, drop a super chat, because super chats get priority, all right? Or if you just want to support what I do, drop a super chat. Don't be shy with them super chats, man. Let's get them cracking. Early birds, what is good? Morph Master J, V Unit in the building. What is up, player? Shout to my V Unit family, by the way. Recently joined the V Unit family. It's amazing. John Elite Exotics BP, what's up, buddy? Trap Talk Patreon member all day, every day. The homie West in the building. Sunshine State Sulfurs. Trap Talk Patreon member all day, every day. Dreeks X. Oh man, you gotta tell me how to say your name, man. Dreek X. Dreek X. Maybe just Dreek, and there's 2 X's on the side. Either way, Trap Talk Patreon member. That's a homie. Appreciate it. Go follow him on Instagram. Morph Valley Reptiles. Trap Talk Patreon member all day, every day. Big Mike, the OG Trap Talk Patreon member. Very honored to have this guy be a longtime supporter, repping Florida all day, every day. Professor Jacob Letterbetter Fetter, aka Civil Serpents in the building. Trap Talk Patreon member all day, every day. It's my dog right here. Appreciate you so much. Heartland Reptiles, the homie Jordan in the building. What is good? Thanks for tapping in. Trap Talk Patreon member all day, every day. The homegirl Chantel in the building. Pacific Rim Serpents. What is good? I also forgot to say, guys, if you want exclusive content on IG, I just started like this new subscription on IG, showing off exclusive content that I'm not going to be showing off to the regular fucking people on Instagram. They're done. I'm doing nothing but heat for my subscribers. Chantel's one of my subscribers on Instagram. If you're on Instagram, subscribe to Trap Talk God 619. You can thank me later, okay? The family's growing. See the top. Thank you so much. I appreciate you. Ethan Hatchery. Oh, by the way, Chantel's also a Trap Talk Patreon member all day, every day. Heavy one. Appreciate you so much. Oh, there's my thing going. All right, so Ethan Hatchery, Trap Talk Patreon member all day, every day. Big time homie, appreciate you. Miller's Menagerie, Trap Talk Patreon member all day, every day. Alex M, looks like a football player, seems like, or maybe lacrosse. Appreciate you being here. Angel, what is up? LA in the building. Reptile Club 310, appreciate you so much. Nick Shanky in the building. Trap Talk Patreon member all day, every day. Aaron from Bosa Balls in the building. V Unit in the house. Piyush Patel in the building. Trap Talk Patreon member all day, every day. Deviant Glass in the building. Trap Talk Patreon member. And we're going to end this in style, okay? Because one of my most favorite podcasts is that hit the reptile game right now. Super down to earth, super genuine, and some real OG advice because he brings some real players onto that show. That's my boy Ray, my favorite reptile podcast. Subscribe right now and don't sleep on him. 5 o'clock Eastern Standard Time every Friday. Don't sleep on it. Guys, you know what? It's time to get going on this. I'm sick and tired of everyone freaking out about DG. What the hell is there to freak out about? Ain't nothing to freak out about. So can we stop freaking out? Episode 353. Ben, Dr. Ben. Morale. Yes, I said it. Morale. Let's go, guys. You know what? I got a special guest tapping in with us too, and you'll see who that is in just a second. But it's game time. Do what you got to do to stay hydrated. Get your mind right. Trap Talk coming at you right now. Choo!
Good.
You ready for do more in the future? Trap Talk podcasts?
Yes.
Man.
Only Trap Talk. Exclusive.
Yes.
Exclusive. Oh.
So stop calling us.
Scrub the spot, get the club to pop. When I come up with the crop, gotta love it or not. I'm hot from the hop to the scrub the spot, get the club to pop. When I come up with the scrub the spot, get the club to pop. When I come up with the scrub the spot, get the club to pop. When I come up with the scrub the spot, get the club to pop. When I come up with everybody, we do it. Everybody, we do it. Do it. Do it.
And we're live with my boy Mark Bailey. Mark Bailey in the building. What's up, buddy?
Doing it, man. Doing it.
Second time on Trap Talk within a week. That's nuts. How did I make that happen? I appreciate that. This is huge.
Yeah, dude, I'm like humbled, man. I don't know what to say.
Well, I could not ask for anyone else better to be a part of this podcast tonight because, talk on the streets, man. So much going on with DG, and tonight we have someone to do his best to kind of calm people down, I guess. But Dr. Ben Morrow, my bad, Morale. I said it right the first time. Moral,
like the moral of the story.
Moral, that's right. Okay, Dr. Ben Morrow, what's up, buddy? Thanks for tapping in with us. How you doing?
Oh, I'm very happy, thankful to be here.
Stoked to have you here, man. Now, one thing why I wanted Mark Bailey to be a part of this is because, I think because of Mark and one other breeder, I just recently got into the DG game. So I'm excited to be a part of it, but goddamn, that's a huge foundation, I feel like, for you, Mark, right? DG is a huge recipe to what you have going on over your collection. Correct me if I'm wrong, Mark.
It's definitely part of the picture. I've worked with it for quite a few years, and it really hasn't been until I'd say the last really 5 or 6 years that it's really become popular. It kind of there were some years there where it faded, so we didn't pursue projects with it. So
yeah, and then Ben, do you go by Ben? Is that can I call you Ben? Is that best?
Yep.
Okay. Ben, you're no stranger to the ball python game either. It's not like you kind of just discovered what it is that we have going on, and then you're into ball pythons. How far back do you and the reptile industry go, or the hobby go?
So keeping reptiles, that started when I was 18. I got my very first ball python from a pet store for $100. Actually, my friend bought it first, and I eventually was able to take it to my house. And 2002 was the first time I bred. I bred a pair of normal ball pythons and a pair of normal boas, common boas, and I got a litter of 19 healthy no slug boas and 8 healthy no slug ball pythons, and I've produced ball python eggs every year since. I've worked with some other species too, some colubrids and many different python species. I was one of the authors on the complete carpet python, the first edition. They just came out with the second, but anyway, I wrote one of the chapters in that. I did my dissertation. I worked with the snake keeper with Dan and Colette Sutherland. I analyzed about 10 years worth of ball python breeding data that Colette had taken. She had written, this is all handwritten. I had to do hours and hours of data entry from this huge pile of papers that she had handwritten out, what order the eggs hatched, the length, width, weight, which one hatched first out of the clutch, all this stuff. And I got to do that for my dissertation. So yeah, keeping, breeding, studying, it's been that way for 20 years for me.
That's awesome. And how did you stumble across, this position now in the industry? I want to say, because it's a very unique but awesome position you are in right now, getting people so excited in the ball python and the reptile industry, because obviously whatever happens with ball pythons will eventually happen for other species. I'm assuming, right?
Oh yeah, we're already working on that right now.
Woo.
So really the seed for it was, as a high school student, I started working in a herpetology lab. I just called the local university and said, hey, I would love to be able to talk to a herpetologist. And thankfully there was one patient enough to talk to a high schooler, and he let me come in and start working in his lab. His name is Joe Mendelson. He's the curator of reptiles at the Atlanta Zoo now. But what he told me was, I remember complaining about classes in college that felt like a waste of time. He said, no matter what you're doing in class, it can be English, can be this, that, whatever. Just think about, I know you, Ben, just think about what you can do to apply that to reptiles, and then you'll be happy. And so ever since then, I've been doing that. I got a degree in molecular biology. I studied genetics and developing genetic tests and doing genetic modifications, mostly in food animal species like pigs and cattle. But in 2011, I got my PhD and got out of academics and got a full-time job. And that's when a friend of mine I'd been talking to online for years, we met in person at the Tinley show in October, Tinley 2011. Sean Christian and I, that's where Rare Genetics Inc. really started. We started talking about being able to offer something that's available to other captive bred animal, industries in various ways, but no one had done anything with reptiles. And so that's where Rare Genetics Inc. really started. And Sean has been a huge part, giving me the faith and the guts to kind of reach out on my own, to let go of a very cushy pharmaceutical job and take a chance at a new venture that I love way more, but is also not as safe.
So this guy Sean, he's a partner of yours. So Sean is more behind the scenes, and you're kind of the front face for all this, is what I'm hearing.
He's definitely a business person. So since he's a teenager, he's run businesses. He's done all kinds of different businesses, and a lot of them were reptile related. That's how we originally met, was on forums and chats and stuff about reptiles back in probably the mid 2000s. So yeah, he's definitely the business side, and he loves reptiles. He's bred lots too, and he can talk reptiles all day. But he's definitely the business. He's the one that figures out how to make this all work. I'm the science. He's the business.
Good dynamic.
I had a great conversation with Sean earlier today. So that was I'm sure that we've met before. I'm sure I met Sean before. Probably met you.
Probably.
Sorry if I don't remember, but yeah, that's a great backstory, and I mean, congratulations to both of you.
Yeah.
This is fantastic. I'm excited about it. There's you guys did a gutsy move on bringing out the DG stuff. Not necessarily prematurely, but we talked about that, and that's quite interesting. And I think in time, once people start learning some of this, and this is tells us what's happening. It's the genetics that we've seen in ball pythons to this point just have been, we haven't had any science behind that. So to have you guys do this and take the time, and I can't imagine the amount of time and work that you've put into it, just repetitive, monotonous, the whole bit, but thanks.
I've been up till 3 or 4 in the morning playing with sheds many times over the last 10 years.
Wow. Yeah. As I'm learning more about it, because we just kept everything so basic before. And there's always questions, not just ball pythons, but boas. I mean, I don't know if any sequencing has been done with boas at all at this point. Are you aware of any?
I know Dr. Warren Booth has done some stuff, especially with parthenogenesis. But the first MORPH stuff, we did an episode probably about a month ago. We would definitely like to figure out a sharp and call strain albino test, so people could get it so that they have just a pure call or pure sharp, have that be a possibility.
I didn't mean to cut you off from the backstory, but it's just a fantastic story you guys have together, and it looks like it's working. So hopefully it continues to evolve, and you make enough money where you can pursue more of this.
So Ben, I have to ask you, Ben, like, what's the day-to-day like now? Obviously there's so much now that everyone knows what is out there as far as possibilities of getting tested. Is it pretty hectic right now? Are you keeping up with all the demands and the sheds coming in and stuff like that? How's the overhead being?
So we had a very painful scale up. So we announced at Tinley last year with Canova, or I guess Clutch, specifically with Clutch and Morph Market. And so for the next 4 months, we were working really hard to scale up, since we had so many more sheds coming in. Now we're scaled up to the point where we're pretty comfortable in handling what's coming through. But the summer, as people are hatching out all their babies in July and August and September, we may need to scale up a little bit more. But I think that we're pretty well set, that if we can have 2 or 3 times as many sheds coming through, I don't think it would slow us down much. We're ready to rock.
So it seems like the real serious time is in kind of basically decoding everything, coming up with finding the genes, and so you have something to go with. So my understanding is you guys took hundreds of normal ball python sheds and started with that as like a baseline, and it all makes sense how all this works. And then you're taking each gene and kind of breaking that down according to the wild caught animal, or you know, the I don't know. There's not too many wild caught ball pythons that come in that might not be genetic, you know, have something going on with them.
Exactly.
You know,
Yeah.
so
So
one to one degree or another.
What we really are able to do is we just took animals that we, you know, either my animals or breeders that I knew were giving me all the information possible. So I could take, like when we were, clown was really the first big one that we worked on. And we took all of these clowns and 100% het clowns, and we knew that was what they were, and then compared that to a bunch of animals that we knew did not have clown in them. So it could be a, you know, pastel stranger monsoon. It could be all kinds of stuff, as long as it's not clown, het clown, or possibly het clown. So we just wanted to have, you know, a couple hundred clowns, a couple hundred non-clown, and we sequence, you know, all of that information, billions and billions and billions of base pairs. And we use bioinformatics software. So it's a branch of science that's fairly new these last about 10 years, where they focus on analyzing gigantic, you know, amounts of data. And so what we do is we take this pool of ones that have clown in it and compare it to the pool of ones that don't have clown. And it just goes through all of those billions of base pairs and said, okay, at this one spot, we see something in these clown and het clown animals, and we never see it in these non-clown animals. And then that's the one that I develop a test for.
And you have to do that with each gene separately to and hope that you can find it.
Exactly.
Yeah.
Sometimes it works great, and sometimes it doesn't work at all.
And then you come up with the curveball of the polygenic DG. So
yes, that was a pain in the butt. And that's another reason why, when we finally got some clarity of why it was such a pain in the butt, we wanted to talk about it, because people have been asking for that for months. So it's nice to finally have a reason why it's been such a pain.
And you know what? I think the biggest shocker is, like, people are probably like mind blown with this new technology, and they thought it was going to be super straightforward, you know, with certain things. You know, they thought, like, perfect, here's my money, tell me what my answers are, you know. But it's not that easy, obviously, because the ball python game isn't that easy when it comes to these morphs, you know. Like, we don't really truly know. Like Mark said, no one's really done deep dive, fucking really looked into this type of shit. So I think this is just the beginning of shocker news on a lot of morphs. But what is somebody supposed to do? Like why the freakouts? And have you even thought about, like, why are people freaking out? Like, do you think why people would want to freak out over something like this? Is it just overall lack of knowledge, you think, or what is it?
Yeah, it's been really interesting. I obviously am not the typical, like, you know, obviously I've focused my life to you know, this kind of genetics information. So I'm not the typical person consuming this information. So it was hard for me to gauge how people would react. What surprised me the most was that first video. Out of that whole video, it seemed like people took the parts that were potentially scary, and that's the only thing they learned and the only thing they remembered. I didn't expect that. Maybe I should have. I'll definitely think more that way from now on. But yeah, I guess it really boils down to, we're scared of things that we don't understand. It's new.
I think that's exactly it.
Yeah, there's a lot of people when banana, you know, when there's all this weird stuff going on with banana, what in the world? And, you know, I remember a lot of breeders talking about that at the time. And is this person really telling the truth? And blah, 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 you know, like all this drama. And, you know, it took some time to figure it out. That one was kind of different, because from the beginning, you know, we knew there was something weird. In this case, we've been breeding it for a long time, and we're learning, oh, this is really close to what we thought it was, but there's something different going on. And so it's been interesting. Certainly, definitely nothing to freak out about, but I have learned a lot in talking to people that are, you know, watching your podcast. And Justin Kabelka, he's worked alongside from the very beginning. I went to his place like a year and a half ago, and he let me just spend all day. I was literally 6-7 hours. We took a break to get lunch, but I was just going through, pulling shed after shed after shed, recording the genetics of it, boom, 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 hour after hour, as many as I could get. But yeah, people like that have experience in the industry and experience explaining things to people that, you know, this is going to be brand new information, that you're much better at gauging that reaction than I am. That's not something I'm good at.
So Dr. Ben,
Mark, hold on. I'm sorry. Hold that thought. Hold that thought. because I know how deep this is going to get. And listen, I gotta look after my green tree python people for a quick second. Okay. My buddy Mike in the building. Shout out to my buddy Mike. Big time concern, not concerned, but what is the news or any updates with green tree python testings? Because I believe he sent some in to you. And I forget, Mike, what the circumstances are, but he's in limbo right now. So what's going on with that?
Yep. So when Sean and I started talking in 2011, this was the exact test that we wanted to provide. We were carpet python breeders,
right,
and we knew how much of a pain it was for green tree python breeders to sex their animals. Yeah. And that was what we wanted to provide as the first test. And it was a pain. And so I ended up providing a colubrid sex determination test. And still to this day, for a little bit, I had a green tree python sex determination test that I worked out, but it was very involved, and I had to have ancestry. I had to have not only the clutch that you wanted to say you had a clutch of eggs, you wanted to get the sex for those babies. I had to have the sire, and I had to have either the grandsire, the paternal grandsire, or a known male offspring from the same sire, because I had to actually track a marker on the Y chromosome. And so I would know if that marker was in a baby, it had to be a male. And so it was very difficult to do. It took a lot of analysis time. And once we announced in October and started, doing everything else, I couldn't do that same type of a test anymore. If I were to force it, it would be, like $300 per test. And, it doesn't fit with what we're doing. So the single shed test is what we've been wanting to have forever. And I still haven't gotten that to work well. We got it to work with ball pythons, so we're close. But all the stuff I learned about ball python sex-specific markers, it doesn't work in green trees. And so we just spent a bunch more money. We sent between 30 and 40 known sex green tree python samples off to get whole genome sequencing done. And so it's another batch of a whole bunch of information. So hopefully this time we can get it. I'll have that data in about a month. So okay, as long as I don't have another surprise, we're hitting it hard with a bunch. That's definitely by far and away, they're probably the group of people the most mad at me, but by far and away, that's the group. That's the test I've spent the most money and time on that still doesn't work.
Relax. Hey, Condor guys, relax off my boy Ben, bro. Relax, okay? Please. He's trying.
I feel bad because I've told him so many times it's close, or I think I'll have it soon. And, but I mean, God's honest truth. I have done everything I can with money and with time, and I just haven't gotten that one to work. But I do think we've hit it hard enough with the sequencing. In about a month, hopefully we'll have something that I can design off of.
All right. Thank you so much. Go ahead, Mark. Sorry about that.
Yeah.
You forgot, didn't you?
Yeah, I did. Of course I did.
Son of a bitch. I knew it. And that's why I didn't want to do it. I was like, he's going to forget.
I remembered it, and then I forgot. So, well, I mean, all good.
We were obviously in depth talking about the it wasn't specific DG that we were just talking about before we got into green tree pythons. I'm sorry. I'm trying to remember here. Yeah, okay, listen, I want to kick this. I want to kick things off with this because at the end of the day, I always cringe when I hear a morph that I really like, and then there's another name for it, right? Like we were just talking about coral glow and banana. Like for me, I was like no one got mad if I got a coral glow and I called it banana. So I fucking called it banana. I don't like that name coral glow. So I called it banana, right? And then just getting into the DG game, I saw this enhancer shit. And I was like, what the fuck is enhancer? And it's like, well, it's sort of the same thing. I'm like, okay, so let me ask you this. If I were to inquire someone's collection who had enhancer and just like the banana coral gene, I was like, I don't fucking like that name. I want to call them DG. And I named them all DG. Can I do that? And they all said no. So I'm like, well, now, and this is before this, what it was, right? But now we're here, now we're talking about DG with a certain level of DG, like DGA, DGB, DGC, right? But then again, are we talking about how we're not allowed to mix those? Like, or is it just for people who want to have the purest DG? This is what we're trying to get at? I'm just trying to wrap my head around this.
So when you had Justin on Thursday, he talked a little bit about how when he has different lines, he keeps that name. So I've definitely run samples from Justin that are labeled as enhancer, and I see the identical DGA, DGB in those animals. What we may see, I can't put into words what I'm seeing in the DNA, but I've done this long enough that
Oh,
Oh man. Now listen, his kids are big time
So-
gamers. Apparently his kids are all over the internet.
There
Oh,
he
here he
is.
is. Back.
Lost it.
Okay.
Okay, sorry. I think that there's a chance there may be like a little bit more DGC, like a higher percent of one of these other genes that we don't have located yet. That's possible that there's a higher percent of that. But my guess is at the end of the day, if we can chase down 3 or 4 or 5, whatever it ends up being, genes that affect desert ghost, I think we'll find that they're in enhancer and desert ghost. It's just maybe a different combination, or maybe one of them is more likely to be homozygous in one, and the other is more likely to have something else be homozygous. That's my guess at this point.
So Dr. Ben, I've seen two different types of desert ghost in my breedings. And I did quite a few breedings that were only desert ghost, no other mutations added to it years ago. And I used to get very clean ones, and I'd get some that were like a little darker.
Can I show you? I have 2 different, and while you're talking, I have 2 different looking DGs, and I want to show them.
Are they just DG, or they have something else in them?
They're both DG het clowns. So does that matter? They're both.
I think the het clown actually does a little something to it. It adds a little, from at least what I see visually. But so the science of this, the DGA, DGB, and C. Now, and correct me if I'm correct me if I'm wrong. With my thinking, is it possible that the non-homozygous animal could appear slightly desert ghost?
Oh yeah, that's like a pastel. Yeah, I mean, if you're heterozygous, you look like a pastel. If you're homozygous, you look like a super pastel. That certainly could be the case with some of these genes that are contributing to that desert ghost look.
This one's in shed, unfortunately, but you could see that this yellow is a lot different than this yellow, even in shed.
Mm-hmm.
And I don't know if these are the 2 you're talking about, Mark. Mark, this is from you, by the way. Okay, I asked you if there's OD in this. You said no, but this is from Levance. So I don't know if you could. I don't know if it's hard for you to see, Mark.
It's hard to see. That's a little dark. Yeah,
and then here.
I would consider that dark. That looks like it has OD.
I told you. Oh my God!
So,
Get out of here.
Well,
but
we'll hopefully have an OD test soon.
I think there might be something going on with that, because we've had all different types of ODs pop. Yeah. Okay. So
Yeah.
we'll learn more from that. But I'm still confused about the question I asked you about DG. Okay. So we have DGA, DGB, and you have to have one on both sides. You have to have both on both sides to show a visual, correct?
That's what we're seeing so far. Yes. Yeah,
Okay,
you have to
so
have
if
at least one mutation at A and at least one mutation at B.
Right. Okay. If you don't, could you still see desert ghost in an offspring?
So far, all I have is what breeders have told me. So they've said, this is a visual DG. This is what I consider a het DG from the pairing it came from. And then animals, they said, do not have DG in their line at all. So they should be normal for DG.
Right. Right.
So far what we've seen is if they say it's a visual DG, it has at least one mutation at A and at B. If it's either heterozygous at one and 0 at the other, or homozygous at one and 0 at the other, it's not been what they call a visual DG so far.
Okay. Because I think it's so muddled up now because there's so many other mutations on, combined with it. How often do you see a DG that's not het for anything or have an incomplete dominant gene? What I used to have to do, and maybe it's just me, but when the desert ghost would hatch, I couldn't tell for sure what was a desert ghost and what was a normal. So I'd raise those, and by several sheds, 4 or 5 sheds, there'd be a pretty good difference. But within those within the differences, there would be like the two types of desert ghosts. So I just
Interesting.
yeah, that's
So one thing that could happen, this is purely speculation. One thing that could happen is maybe you had breeders that were homozygous for like A and B, but C, one of them was negative for it and the other one was het. And so every time it got that third dose, it got a different look, whether it made it darker or lighter. I mean, who knows? But you have to remember with
that C
polygenic.
can go ahead.
Yeah, with polygenic, each of those genes could be acting differently. One could be recessive, one could be incomplete dominant, the other one could be dominant. You know, it's not like that trait has to be recessive, you know, dominant or incomplete dominant. It's each gene in that, you know, in that group, that polygenic group. It's the same with eye color. We have some genes that affect eye color that are dominant and some that are not. You know, they're recessive. So it's the same thing. You could very well have your animals are mostly getting A and B, whether homozygous or heterozygous, but what's making the difference is what they get at C. And that might be what you're seeing. And about half of them are getting it, half of them aren't.
Half of them aren't. And statistically, that would be the way C could potentially work, is 50-50 on, okay.
Could. Yep.
Yeah.
That's very possible. It could also be, which, you know, I have learned to be careful to not say things that people freak out about.
I'm not saying I'm not going to hold you to anything here, you know, until the
what could also
comes
be
out,
is there's three other genes, you know, not just one. And as long as you're heterozygous for one or two of those, you'll see it. But if you're not heterozygous for any of them, you won't. You know, there could be all kinds of different combinations. Once it's a polygenic trait, it's a lot harder to nail down. We're lucky we've got two of the most important pieces. I think C is worth chasing. I think at least those three I'm going to want to have in a test. But yeah, it can definitely get complicated what is going to lead to a certain look at the end of the day. But
because you're looking at the science end of it, the genetics that you're seeing on your sequencing. We're looking at the actual offspring.
Yep.
You know, those are, so it's hard for you to know without knowing exactly what these things look like.
Yeah.
Okay.
Yeah. So that will be really good over time to see. And in that episode that you all did with Justin and Summer, that was really cool to see some of those that had been genotyped, and, you know, you can see the animal and then they tell you what the genotype was. And as we do, you know, more and more like that, and people post pictures and videos showing, especially if they're from the same clutch, and they have similar genes, but they have different DG, you know, genotypes. It'll be really cool to see those side by side. Maybe one will always be darker than the other, and that will kind of explain what it was that you saw back in the day.
Yeah, I think we need to see full clutches of only Desert Ghost.
That would be the best.
From het. I did a lot of het-to-het breedings, and a lot of my het-to-het breedings were very high percentage DGs. So, you know, but I have made what I consider to be both. And yeah, this is all interesting. I clearly haven't got my head wrapped around it all, but I'm going to keep studying this until I do. And I think that's the trick for everybody.
Oh yeah,
you know,
I'm learning too.
Right. Well,
learning could be fun, guys. Like, let's make this fun. It's all about learning, right?
Absolutely. I mean, that's what we're here for. You know, I mean, we're, you know, I wouldn't be still doing this if I wasn't learning something every day. You know, it's.
I'm gonna take this. I'm gonna take the super chat real quick from my buddy over at Covert Reptiles. So, can polygenic be influenced by locality or the environment they came from?
It certainly can. One really good example, I had Dr. Travis Wyman on the episode we just released a couple hours ago today. One example he brought up of, you know, we were just talking about interesting things we know happen in other species and we haven't seen in reptiles yet, but may, with Siamese cats, that albino look, that's temperature dependent. So the parts of the animal that are the warmest have a different coloration than like the tip of the tail, the tip of the ears, you know, the tips of the paws or whatever. So yeah, in that case, that certainly happens. There are other things that we did an episode on the piebald gecko, or the piebald anole lizard that the researchers in Georgia and Canada worked together on. So they took the code that they learned, made piebald ball pythons, and they used CRISPR to put that code into an anole lizard. It's the same group that made the albino anole a little bit before that. But in that case, that it's the same kind of a thing. The paws and the snout, like the parts of the animal that are a little cooler, the proteins interact differently, and so you get a different coloration. In that case, it's more of a white color and a little translucent, whereas the rest of the animal, where the temperature is a little warmer, has more of a normal coloration. So yeah, that certainly can be for some genes. Whether that would be the case for desert ghosts, we don't have any evidence of that yet, but you know, that stuff happens for sure.
This stuff is cool. I remember now, MJ. So, Dr. Ben, what I had brought up earlier, your YouTube videos are fantastic.
Thank you.
I've only watched a couple of them, but I think I want to watch all of them. I mean, I think that can provide all of us a good learning tool for genetics. And you know, I encourage you to, well, maybe I'm being selfish, but I encourage you to keep going with that. You know, I can get a free education without having to pay. You know, so.
Well, Kayla and I both made a pact with each other that we will do 52 episodes. We'll do a year, and whether it's going well or going bad, either way, we're doing 52 episodes. And at the end of that year, we'll have a talk. And if we want to keep going, we'll keep going. If not, but yeah, we're already in it. We're already, you know, dedicated. We'll get those 52 in.
Yeah, guys, head over to YouTube right now. I'm obviously already here, but subscribe. Rare Genetics Inc. Mark Bailey likes it. You're going to want to go ahead and take advantage of it because, Mark, I love how long you've been in the game, and you could still not get enough when it comes to learning new shit. You're all about learning new shit, bro. And I think that's kind of
every day, man. Every day.
Yeah, man. It's very inspiring. But also, let me ask you this, man. It seems like you're not spreading yourself thin, but with all this shit that you have going on top of social media stuff, man, do you feel overwhelmed at all? Like it does catch up, I'm assuming.
Yeah, for me, I feel kind of sad that I can't do all of the things every day. Like these last couple of days, or probably 3 or 4, I haven't been able to answer questions. I did 2 or 3 days straight. Well, the day after the episode came out, that night I stayed up till like 4 or 5 in the morning. I slept for like 3 or 4 hours and got up, and I literally answered questions until like 8 at night. I didn't do any of my analysis. Wow. I had a bunch of tests I was supposed to analyze and get out to people, but I felt like it was pretty critical to answer questions that first couple of days. But so yeah, these last 3 or 4 days, I haven't answered questions. But the good thing is, you know, some people that asked questions early on, I've answered, and similar questions are being asked, and some of those people are answering for me. So that's great.
I was going to say also, Ben, you know, I know you have experience, or you kind of came into this breeding on your own. What are you currently working with? Like, you have your own stuff that you have in your own collection that that this stuff can benefit you from as well?
Oh yeah. I've done, so I had a male that was a clown posset pied combo, and I had a male that was a pied posset, or a clown posset pied combo. Anyway, one of each. But I was able to test those, and last year I kept the ones that tested positive, and I got clutches from both of them. So I made some triple hets. I wouldn't have even done that pairing, but since I knew I could test, I'd made triple hets from both of those males, and so that was really nice.
Let's see if Mark could predict what's in these right here. What do you think's going on here, Mark?
Oh God, I think champagne, maybe.
You're right.
Yeah. I almost want to say Mojave.
No, let's see. Yeah, no blue-eyed Lucy complex genes at all.
This is cool looking, man. This is like a sherbet.
Axanthic?
No axanthic.
Hypo?
Yep, it's a hypo.
Oh,
I was gonna say that, but that's hindsight.
And there's, I don't know. I don't want to say pied. It could just be a pied marker. Was that a pied?
I think that's pied marker. Yeah.
That's the champagne talking.
Oh, champagne does that? Okay.
Yeah, I had some OD champagnes that did that fairly extreme.
Yep, so, this has got OD.
OD champagne hypo?
Mm-hmm.
So it's an OD champagne hypo. It's also black pastel and het rate axanthic.
Holy shit.
So Dr. Ben, I got a question about that. Okay, champagne, black pastel. there, we have for all the visual genes and mutations that we've seen in ball pythons, the majority of them have been strong. Some of them, including desert ghost, I've always thought to be a very strong gene. Everything desert ghost does fantastic. I mean, they get big, they lay big clutches, the whole bit. Champagne has like a little head thing. The spider thing has a head thing. You can't breed two spiders together. You can't breed two champagnes together. You wind up with really neurologically corrupted animals.
Hey, I got a question with that, Mark, and you too, Ben. Let me ask you this, because I heard, like, for instance, you can't do spot nose and spot nose, right?
You can, but I think some of the first ones were showing up dead.
So if that's the case, can you put two morphs that don't do good as a super to another? Like, can you do like let's just say a cypress to a spot nose? That would be fine, right? Like that's.
You mean something that's allelic?
Yeah. And that should be fine. Is that like a way to kind of dance around that or something,
I know that some of them are still not good. I haven't worked with cypress at all, but actually blackhead in spiders, the opposite. Those are allelic, and you can definitely make blackhead spiders, and they are very hardy.
That's interesting.
No wobble.
Wait, so this is something that you're going to have to work on down the line at some point, because that's the stuff that really has me scratching my head. Why do we have some that are so good and some that just aren't?
So specifically with spider, there's been some really cool research done. They went in and took some really good high quality electrographs. So like they're using a microscope, going in, looking at the physiology, the neurological physiology of spider ball pythons compared to normals. And actually back in the day when I was a grad student, there was a chicken researcher, and I was always talking about snakes, and it's talking about spider and the wobble and all that stuff. He's like, oh, we have that in chickens too. And he talked about it is exactly the same kind of a thing.
Birds and reptiles. Yeah.
Yep. So what happens with spider, most likely, if it's what's happening with chickens, which makes perfect sense to me, is the cells that move along the neural crest during embryonic development. There are melanocytes that travel together with neurons. And so what that mutation does is it messes up where those neural cells go, and those melanocytes are kind of going the same way. And so that's why you get a change in pattern, but you also get a change in how the quality of the neural network for that individual. And so that's why you can't really outbreed and get rid of that neurological problem with spiders, because those cells during embryonic development are traveling together to make that neural network and to make the pattern. And we like the cool, crazy looking pattern. At the same time, you're also getting a crazy neural network.
Almost sounds polygenic. That's crazy. I think I followed you there, but now my brain's just going every which way. So
I'm glad you remember what you had to say. That's important.
That paper is pretty cool. I'll have to try to remember to talk about that and provide a link for people. But if you just look up spider ball python in the scientific literature, there's not very many papers.
So in your description of that, you made a comment about the genetic pattern changing gene. Are there color and pattern genes together? Because I think Desert Ghost is a slight pattern. It affects the pattern slightly.
Yeah. So that certainly has happened. A good way to think about genes. So we, as humans, we have like 35,000 genes that do everything, to make a skin cell, to make a heart cell, to make our whole body. All that information is coded by 35,000 genes. They're doing way more than that many functions. So any gene that you ever study has more than one thing that it does. So it's very common that a gene that affects color will also do something else. It could be pattern. It could be, some of them will affect how the heart forms during embryonic development, or it'll do this or that. And so it's almost never happens that a gene only has one function.
Okay. Oh, that explains that pretty simply. Yeah.
Guys, if you don't mind, guys, 145 people in the building. I appreciate you. Let's get the likes up. This is an amazing podcast. Very informational. Mark's killing it. Ben's killing it. Dr. Ben. Can I call you Dr. Ben? Because I think that's a really cool name. I've never.
Sure.
Respect, Dr. Ben.
I have too many friends that are doctors, so this is pretty cool. Dr. Ben, I have to ask you, I mean, any thoughts of what's next on this hit list? And when I mean hit list, like DG was probably like, what a discovery to figure this out. Any other morph out there that you guys are curious about that you maybe want to like, I think this is next. Let's figure this one out.
Yeah. Well, first of all, I'm curious about all of them. I want to do all of them.
Right.
Yeah, but we do know what's next, because all of this takes a lot of time and a lot of money. We're talking, tens of thousands of dollars all day, all the time. So the next one coming the next big one that I think will affect a lot of people is Sunset. So that one I expect like within the month, 4 to 6 weeks, but if I get lucky, faster than that. So Sunset's coming, VPI Xanthic and TSK Xanthic. We have Monarch that's really close. So those 4 are probably out of the big ones, those are close. But I'm also working kind of on the side, things like Spider and Blackhead and Red Stripe, Orange Dream, Pastel. Those are kind of things that I keep pushing forward. And so just a couple weeks ago, we had both Inchy and Black Pastel pop out. We got those figured out. And those were kind of these same ones that I've had kind of as a back burner. Not necessarily something that a bunch of people are going to, want for a single gene test, but people that have one pet ball python, they want to know what it has, and makes that panel more valuable to them, because right now, I think we have like close to 15, and within the next month, we should be up to 25 different ball python morphs that we can test for.
Are those two allelic?
Which Inchy and Black Pastel?
Yeah.
Yes.
Okay, and cinnamon is too. So.
Yep.
Right. Yeah, and we're finding that there seems to be a decent number of these incomplete dominant genes that are allelic.
Yeah.
Why would that be?
That was super interesting with the yellow belly complex paper that Dr. Sadel published. Mm-hmm. I just kind of assumed that like gravel and asphalt were probably going to be the same thing. I wouldn't have thought that it would be literally a different, actual mutation.
I've worked with those mutations from the very get-go, so that's been a fascinating thing to me.
Yeah.
But yeah, how that's worked.
Genetically, it's the same gene that's mutated, but it's mutated at a different place. Mm-hmm. And it makes a little bit of a difference in what we visually see, enough of a difference that breeders could tell there's a difference between gravels and asphalts, where, for me, I've never worked with it. I had a gravel female for a little while, but I've never produced any gravels or asphalts or supers or anything like that. And so I was really surprised that there was literally a DNA code difference between them.
Why would they all be on that same area, though?
I think that we just, people out in Africa catching these wild, or
now,
maybe they're getting a bunch of them, and what comes in, what we're sifting through. There's just something that caught our eye, and any mutation to that gene changed it enough. They're like, oh, that looks cool. And so it's more that we are selecting for the ones that look cool, and they happen to be on that same gene. There's going to be random mutations everywhere. And the ones that are really bad for the health of the animal, those animals die. Either they're not born, they die in the egg, or they don't do well once they're born. So the few that make it through, that's just
nature's attempt at getting better.
Yeah. Yep. The ones that get through are ones that can go through embryonic development, and they can, eat and grow. And I think that with the case of the yellow belly complex, it was just different enough that we could pick that up by eye. And, now we know. I just want,
that with the ball pythons is geographically spread out over Africa that they are. I just wonder if all those came from the same locality, or that's probably something we'll never know,
So I can tell you, from a geneticist's standpoint, that it would be extremely unlikely that any of them came from multiple individuals except for yellow belly. Yellow belly, there's two different codes, -1 and a -17, that both make a yellow belly that's indistinguishable. So that one, probably there's two originations, but gravel is likely a single, spark is likely a single, specter, asphalt, those are all likely single instances where that mutation happened in the wild, and then it spread.
Yeah.
God, that's a trip. Yeah.
Now, I don't know that for sure, but the chance of the
right.
exact same mutation happening randomly is pretty close to zero.
Right. And speaking of percentages on while I'm thinking about it, the whole thing about the visual DG to a visual DG not making a homozygous animal, what would the percentages of that be?
It would depend on the genotype of your adults. So, I think that those of you that have bred them for many years know better than me, but from talking to breeders, it seems like it's extremely low percentage. When Justin was on the other day, this same podcast, he brought up that one individual that was from a DG to DG pairing, but he said now that it's had a few sheds, has a little bit of size, he wouldn't have called it a DG. Looking at it now. And another way I've heard him say it before is if that popped out of another clutch, I wouldn't say, oh wow, that's a DG. But since it's a DG to DG clutch, you're like, oh, right, it has to be DG.
But those are they still have other genes with them that kind of muddle the whole picture there.
Definitely makes it more complicated.
Yeah. Well, we'll have to pay attention to the regular DG clutches, I guess.
Say that again, Mark.
Yeah. Well, I know what Yeah.
Shout out to Dennis West. I'm gonna put a couple super chats here on the screen. Dennis West has to say, does AAB BBA and AABB DG animals need the C gene to be visual, like it does for AB needing C to be visual? Wow. Fuck.
So to hopefully simplify, I think I'm understanding the question correctly. To try to simplify that, do you need a mutation at 3 places? So we're saying so far we've seen you have to have it. Every visual DG we've looked at has a mutation at A and at B, at least one. And I think he's asking, do at least in some combinations, do you have to have it at C as well? And yeah, we are seeing that it looks like C is, especially if it's het for DGA and het for DGB, we definitely see some of those are clear visual DG and some of them are not. And the breeder's calling that a het, not a visual DG. So definitely in the DGA het, DGB het situation, that C definitely makes a difference in whether it's a visual desert ghost or not. But since we don't know where C is, we can't test it yet. It's still a little bit of a guess, but it's a very educated guess at this point that C will be needed for at least some gene combinations to be able to make it be visual.
Respect to that question. Here we go.
That's why we want to know where C is.
Yeah. The professor in the building, Civil Serpents, has to say, Dr. Ben, when will we be able to submit our DG sheds for the actual DGA DGB test?
Probably 3 weeks ago.
3 weeks ago.
Yeah.
So he's not informed. Now he is, but he wasn't informed, I guess.
One potential reason why is I know MORPH market has been a little slow to get it added. So through Clutch, that's been available for a while now. And as soon as I had B, the first few that came through Clutch, we just tested for DGA, and that's why Clutch called it an elimination test, because at that point I didn't know for sure what the second one was, or if there was a second. I hadn't tested that yet. So the very first people that got a DG test from us, that was just DGA, and that's why we called it an elimination test and only 85% accurate, because I knew it wasn't telling the whole story. But yeah, for the last 3 weeks, so the last 2 runs that have gone through, people that had a DG test in there, whether it was through Clutch or MORPH Market, they got DGA and DGB results. So if MORPH Market still doesn't have it added, what you can do, and you want to test your shed for DG right now through Morph Market, just pay for albino and then just write on the bag itself, say, please test DG in all caps instead, so that we know, don't worry about albino. That's just to get it, paid for and in the system. And then when we report the results back to you, we'll give you
Always something.
Yeah, it's okay. He might kick back in.
Sorry, where did I cut out?
Go ahead.
No.
Well, you were just finishing up after the albino on the bag and all that.
Okay, sorry, I don't know why it's cutting out so much, but yeah, you write that on the bag, and we will make sure that we run it for DG instead. And in the notes, every time we give you your shed results back, there's a notes section, and we'll put in the notes DGA header homozygous or negative, DGB het homozygous or negative. So you'll get your results for A and B.
Can we poke the fire a little bit? I want to poke the fire if that's cool with you guys. Just
Sure.
I got a fire poker. Mark, can't wait for this one.
Poke it.
Sunset.
Okay.
I want to talk about Sunset a little bit, okay?
Yeah.
Sunset's been it's been through a lot, I feel like, the last year. And what I mean by that, it's like, we're talking about people who are mainly freaking out about DG, who don't really have DG in their projects. Like they're just kind of on the outside looking in, and they're just kind of expressing their feelings, which is valid. Go ahead, right? But I feel like it's the same, and this is me. I'm a culprit with this. I have my doubts about Sunset, unfortunately. I think there's the future for Sunset at some point, but right now, I don't think it's anytime soon. For me, as far as seeing something that's going to be like, holy shit, let's stop what we're doing and let's look at this snake. But also, I don't have any Sunset, so it's easy for me to say, right? I want to know where you feel, Dr. Ben, with Sunset, and obviously, Mark, I want you to tell the people what you feel about Sunset and the future for it. So let's talk about Sunset.
So for me personally, as a breeder, I don't have any Sunset at all. So I'm kind of in the same camp as you. It's not something that I've obsessed about. The genes that I am really excited about, I've obsessed about, and think of all these combinations I think will look cool and stuff like that. That's not something I've done with Sunset yet. I've seen 2 or 3 that looked really interesting. I was like, oh, well, maybe I will think about getting Sunset at some point. Majority of them, they're okay, but there are some combinations people are making now that get my attention.
Well, MJ, if you remember, well, you don't remember, but we've talked enough about DG was very quiet at the beginning too, and we see what happened with that. So never say never. Yeah. I do not own a Sunset myself. I don't have plans of going to get one tomorrow or anything. Just because.
What about super cheap? Like, what if someone's like, "Man, I got an opportunity for you, man," and like you wouldn't.
Maybe that's been done. I don't. And it's not about the money. If I wanted it, I'd go buy it or whatever. But I've kind of got my hands full with what I've got. I'm not looking to make every mutation that's out there and put that in. Now, Dr. Ben, in regards to Sunset, you mentioned that you had been doing some work on that. And a few others. Are you seeing anything else that looks, that might be polygenic or?
No, not so far. Weird? So when we do those comparisons with the, in this case, Sunset and no Sunset, and we have these whole genome sequence, we lay out all of the chromosomes. And then it shows like which chromosome will just get one big old peak. So like with Sunset, with Clown, with Hypo, with, all these others that we've done so far, we just get one big peak. And that's where we look to see where there are changes that may actually be the cause of Sunset. But when we went and did DG, we saw 2 peaks. So, instead of just this one, we got 2 obvious places that we needed to look. And at first we thought, well, a lot of people have pastel desert ghosts. Maybe we're just seeing a peak for desert ghost and a peak for pastel. But when we did more sequencing, we saw those peaks still, and we knew that we were, taking the ones out that had pastel. So that wasn't explaining it. So yeah, so far we haven't had a double peaking gene like desert ghost. And after seeing your podcast on Thursday, that made me, looking at the pictures with Justin that Justin put up and Summer, made me more interested in making sure I find that third gene. And so I see another third really small peak, and I've already designed stuff and ordered, and so I'll have some data on that actually the end of this week. It might be complete garbage, but there was a little bit of noise there that could be real. So we'll see.
Okay, interesting.
Yeah, but yeah, the other ones we've looked at.
You just set yourself up, or everybody's gonna be looking at you by the end of the
week. Well, if you don't hear anything, that means that it was garbage.
Yeah.
If I see something, I'll talk about it.
To switch the subject from DG for a second, on one of your YouTube videos, you were talking about hypo.
Mm-hmm.
You're only seeing one type of hypo.
That's all that. So Dr. Sadell did the majority of the work on that. The same person did the yellow belly paper and the albino and lavender albino paper. So what she, and once again, she's in the same boat.
Is she in Virginia as well?
She's actually at Eastern Michigan University.
Okay.
So, but yeah, she is relying on how people describe. So she had people send in ghosts, hypos, and orange ghosts. And that's how they had them labeled, and they're all identical.
There's non-compatible ones out there.
Yeah,
that's what we've heard so far.
Yeah,
and I've told people have brought that up, and I said, send them to me, and I'll run them.
Mm-hmm.
You know, I'll run them for free. If you've got 4 or 5, you can show me pictures, and yourself, hey, these are some kind of hypo. I would love to run it and get a negative,
right?
And then I can have something to chase, but. I've heard that for years, like the citrus hypos.
Citrus is
ghosts,
one.
And like a green ghost or something like that.
There was another one. I think there was at least 3 that I know of. And I call the common one is the one that seems to be most compatible. And I haven't heard of it. I haven't heard of anybody striking out in a while. And I just wonder if that's kind of settled down enough, but it'll pop up. They'll pop up at some point.
If somebody has them and they're pretty sure they're not compatible, or they just think they might not be compatible from what the breeder said that they got them from, I'll happily run 5 or a dozen or whatever for free, just to be able to see if there's another hypo gene to go after. I would love that, and I'm sure Dr. Sadell would love that too.
Right. Yeah. If anybody's got some, send those in. That's I used to have the citrus line. And I that was years ago. I got sold it off somewhere. I don't remember.
How much retic stuff have you gotten request wise? Like, because I gotta tell you, I'm pretty passionate about my super dwarf retics and whatnot. But then there's also a gene that's very questionable. It seems like in projects called anery. And it seems like so many people have believed they had anery, but it turns out that they don't. So I'm curious if anything like that come your way yet. And any kind of update with the retics as far as having be a part of what we're talking about here?
We've had several people ask for retic tests, even just a sex determination test. Some people said that would be helpful to them. And every time I say, send us sheds. So we have, like we talked about earlier, we have to have material to sequence to be able to develop the test. So maybe you all on Trap Talk tonight, maybe you'll be the ones to get this going. Because I've had a few Burmese python sheds come in. I've had a few blood python sheds come in. I don't think I've had any retic sheds come in at all.
Ooh, I'll send some in for sure. Because I got like 3 holdbacks that I was told to have anery in it. And I would love to confirm that. So I can send that in. Oh yeah.
It'll take a while.
MJ, maybe Dr. Ben can come up with a dwarf test.
Yep.
Ooh, dude.
I talked to a couple people about that, and I said, all I need is sheds.
So what we're doing is kind of confirming the locality trait, right? Or like the actual, so if you have a 100% pure Calatoa, you would be able to determine if there's how much percentage of Calatoa is actually in each snake.
Yeah. So that's a potential test. So what we can do.
Yeah, go ahead.
Sorry, what was the last thing you said?
No, I said no. I'm sorry. I was talking out loud. This could change everything for the SD game. So continue. I'm sorry.
So what's possible at this point? We can decide what kind of a test would be the most helpful to breeders. And like I said, I've talked to some bigger retic breeders about this, and there's some excitement about it, but no one's sent me sheds yet. But we can choose. So if what's most important is a locality test, we could do whole genome sequencing and get samples that we believe are as pure as possible. I don't know that we could for sure have 100% pure unless somebody can get a permit to go and collect them in these awesome places that are probably kind of dangerous. But so yeah, if locality is the most important, we could design a test to do that. But I think what would be easier would be to design a test. We could sequence a bunch of animals where we know what their adult length is, or we could say like their length at, you know, so many grams, or we could say their length at 5 years old, or whatever you want to say it. But we could say animals that are 8 feet and longer as adults versus animals that are 8 feet and shorter as adults. And we could come up with a test that would tell you when you breed this male, his offspring have this % chance of being below 8 feet. That's a kind of test and for my PhD, I worked with cattle genetics. I taught a breeding and genetics class, and they have what's called estimated progeny differences. And so for people that have milk cattle, you can buy straws of semen to do artificial insemination. You can buy these really expensive bowls. Instead of buying the bowl, you can just get that semen and do artificial insemination, and it will tell you what % chance those females will be able to give milk, you know, reproduce, give milk sooner, which ones will give more milk, you know, that kind of stuff you can do. We can definitely design that kind of a test. It's been done many times in other species, but I just have to have those sheds. So if I had the simplest one in my mind would be a group of sheds where the adult is, like I said, it could be 10 feet and higher, 8 feet and higher, whatever you want that line to be. And as long as we can trust that people are giving us sheds from animals as such, and then the ones that are less than that, and we could provide a test that would tell you what % chance offspring from that individual. And then if you breed 2 of them together that are a high probability of being below 8 feet, obviously most of the babies are going to be below 8 feet.
You want to know why else this is probably super like next level is because there is a lot of suspect dwarf, super dwarf stuff in a lot of people's collection. So I'm so like, you know me, I'm so excited to send you stuff. I'm curious what big breeders or middle breeders or whatever are like, nah, I don't want to send my stuff in.
Oh, don't do that to him, man. He's a doctor.
Well, no one's told me they don't want to. Everyone's always excited to talk about it.
I would hope so,
like,
but I'm saying, go ahead.
I always say, yeah, send me sheds. They're like, yeah, we'll do that. And then some people do, and then some people don't. And it might be just because of my bias since I breed ball pythons and I talk to ball python breeders that I've gotten lots of ball python sheds. And like I said, I went to Justin's, but I would happily, Sean is in Texas. He would happily go to Oklahoma. There's some, you know, been some big python breeders and, you know, in Texas and near Texas. And anyone near me, I'll happily drive 3 or 4 or 5, 6 hours and just spend all day collecting sheds. I would happily do it. Sean will happily do it. We just have to get those sheds. And like I said, some people come through and some people don't. And I can only go forward with the ones that go through with it.
Some might be afraid of the result, you know?
Yep.
Saying, I mean, there's people who've been in the retic game for years. And like, you know, think about like you having something that you thought you had for this long. And it's like, fuck, do I even want to find out that reality? And it's like, some people are probably innocent. They just bought the animals as is and they don't know. But then, you know, it's like, fuck, it's like the Maury channel, like the results are in.
Well, and then the skepticism of science, you know, you've got that end of it as well. So, you know,
you do definitely have to have a group of people that do honestly want to have the answer. And then, like you just said, Mark, you know, you have to be able to trust that the person designing the test actually knows what they're doing. I have perfect confidence in that because I've seen it in many other species, the same type of a test. And it was very helpful. Like if you're going to have a beef cattle operation or a dairy cattle operation, and you're going to live off of that, you have to be doing this kind of, you know, about EPDs, you know, about the numbers, you know, which ones matter the most. Doesn't matter if you're, you know, backwoods, middle of nowhere, no education, you know, no formal education. If that's how you're earning your living, you sure as hell know what EPDs are. So
right.
I know it'll work in reptiles too. That people have to get those samples in for us to start that.
Hey, Mark, you
Yeah.
brought up a point when you spoke to Sean, you know, and I'm not saying you spoke to Sean about this, but I heard come out your mouth that, you know, are we prematuring this DG testing right now? And you know, you have some people speculating that, like, you know, is this a money grab? Like, and I'm just telling you, like, from the outsiders, you know, saying this, like, you know, maybe they just need, maybe they're putting not pressure, but like, what's your answer to this DG test, like being done now and just waiting to do it when it's more accurate, I guess?
You asking me?
No, Ben.
Yeah.
Oh, me. Okay.
Yeah.
So you're just saying that Mark brought it. I thought you were asking.
I could speak to that too, but
that's what.
ahead.
Yeah. I was bringing up what he mentioned, and so
Yep,
and that
yep.
obviously that's obviously
Gotcha.
you. Yeah. I'm sorry about that.
Yeah. Yeah. So, if you want to wait to do it, that certainly makes perfect sense. You know, no problem with me. I definitely will test all the ones that I have. Not that I have very many. That's a new project for me. I haven't produced any, but I will be this year, either this year or next. I might get a clutch or two this year, but anyway, I mean, if you want to wait, you know, that certainly makes sense, and that's, you know, perfectly understandable. And will it be more accurate later? Yeah. You may miss out on an opportunity to, you know, do a better job choosing your holdbacks, or choosing pairings that you're going to do. I would imagine Justin's done about 100 animals, I think pretty close to it, if not over that now, specifically for DG. And I would bet that that there will be some decisions that will be affected by, you know, that genotype that he sees in the breeders, or which ones he's going to hold back, because, you know, you may not increase your odds by a ton, but if you're producing enough of them, and, you know, obviously you guys know, if you have one come out a certain combo and it's a really nice DG on top of that, you know, that's going to sell much faster and at a higher dollar. So, you know, it just depends on where you are and how excited you are about implementing this. If you don't want to implement it and you want to keep doing what you've been doing, it's been working and you're happy with that, that definitely makes sense.
Yeah.
I think this gives us an opportunity. If we want to test and know exactly what we have, we can predict our results a little bit better than we have been.
Yeah.
So, you know, it's not a necessary thing, but, you know, depending on what level of breeder what level of breeding you're doing, it could make a lot of sense.
Yeah. Now, for anyone who's wondering, because I do have questions, you know, as far as questions on how to get in touch, or the address to send in the sheds, is Morph Marking Clutch the only way where they could kind of do this, or what can they go straight to your website, or what's the best way to kind of see how they could get involved in this?
So there's two different ways to send sheds in. So if you're purchasing a test that's already developed, then you would go through Morph Market or Clutch. But if you want to be a part of what we were talking about, the next retic test, or, you know, sex determination for another species or whatever, if you go to our website and the menu, there's a help us. And if you go to our Instagram or Facebook, we even have a screenshot video that shows you how to do this. But yeah, you just go to the menu, help us, and it will give you a very brief description on how to, yeah, that'd be perfect. Bring it up. But yeah, you go to the help us, it will show you it'll tell you very briefly how to collect a shed. So you just need a piece about the size of a quarter to a silver dollar. We don't need a big piece. And you want it to be, the most important part about getting shed samples is you do not want them to be in a wet, moist, or humid environment for very long. The water is what will destroy the DNA. And so if you're going to collect a shed from an individual, once they go blue and come out of the blue, you know they're within a day or two of shedding. I check those every morning and every night. So it should be within, 6 to 12 hours that I pull that shed out. And if they shed right into the water bowl and there's crap in there and it looks nasty, that's not going to work. Just throw it away and wait for the next one. But yeah, just get the shed out, find the flattest, most clean, prettiest piece that's like the size of a silver dollar to a quarter, something like that. We can actually run like 30 tests on a piece that big. That's plenty for us. We can go back to it multiple times. I have sheds in Ziploc bags that I pulled and collected, you know, properly 5 years ago that are sitting in my basement in a Ziploc bag, and they still run just fine. So that's the biggest thing, is don't let it sit wet for very long. And then, like I said, we just need a piece size of a quarter to a silver dollar. And then at that link, it tells you the address to mail to. And so if you're putting those in fairly small Ziplocs, you can usually get 2 or 3, maybe even 4 in a normal envelope, and it's just one stamp. So you don't need to, you know, be sending priority boxes or anything like that. Just go ahead, small pieces and small Ziplocs, put 2 or 3 in an envelope, one stamp, send it to us.
Bitchin'. You got anything, Mark? So, Dr. Yeah. So, Dr. Ben, I have to ask you, with everything that is happening right now, and the fact that you've been a part of, was it the Morelia? What book did you just say that you wrote a chapter for?
Carpet Python, the complete Carpet Python.
So, shout out to Nick Mutton. You know Nick Mutton, obviously, right?
Yep.
Yeah. I still need to bring him on the show. Brandon, help me set that up, please. But anyways, I got to ask you, any works with maybe writing a book on this? Just to kind of making it more
in his spare time.
I mean, obviously not tomorrow, but I'm just saying, as things progress, as things get more figured out, more dialed in, like the future could possibly hold a book for people to kind of get their hands on. Because, I mean, think about this industry is still growing, no matter what sales are like. Industry is still growing by the day, and we want to make this as easy as possible, but then yet, like, understand that there is a lot of learning to this,
Yeah.
But I gotta tell you, some people just want to have fun, and not that they're lazy to learn, but sometimes they have so much going on with their day where it's like, fuck, I ain't got time to sit here and fucking research all this and do all these test sheds and what the hell is this clutch app? All this stuff is coming up, and obviously it's like, that really, you gotta ask yourself, how professional do I want to be at this, right? I don't even know where I was going with that, to be honest. I just feel like a book would be awesome someday. And like I said, because this is interesting, right? So many people come in for the love of the animal, but then now they become all geeky and they want to know more. I'm one of those. I didn't even know what the fuck a codom or recessive was. After like having 60 ball pythons, I figured it out. That's crazy. And that's because I didn't ask questions. I didn't care. I didn't want to make friends in this. I'm like, I just want to do my thing, man. Leave me alone. And as I was trying to figure it out on my own, that's when I ran into problems. And so, and I feel like this, like, if the future holds a book, I think that would do really well. I feel like, I don't know, call me crazy. Yeah.
We've kind of talked about, with Sean, and then also with like, MORPH market partners and clutch partners, what would be the most effective way to help people learn. And it seems like social media and YouTube was probably, videos and, simple, like the slide deck that Summer put together for Desert Ghost, a visual way to make it as simple as possible. Felt like that's probably the best use of our resources right now. But yeah, I mean, I've written a dissertation. I've written, chapters of books. I haven't done a whole book on my own, but that's certainly something that I would enjoy doing. But yeah, it'll be a few years out. I need to get a lot of this stuff done because I'm very excited. I think about it all the time. I got to get through a whole bunch of these morphs, and I really love the sex chromosome, sex determination test development as well. That's a lot of fun. I want to do that for blue tongues and for Gila monsters and for monitor lizards and some of these other species that'll be really helpful for. But yeah, once I get a whole bunch of reptile tests developed and running and helping breeders, then sit back and spend the time writing instead of designing new tests. I would dig that. That'd be fun.
Oh yeah. You have a wrap up question for him, Mark? And then or I could ask a wrap up question if you want to get one in your head while I ask him one. Yeah. Can you think of one while I ask him one, Mark?
Well, I was going to expand on what you're talking about, but
okay,
before that, the ball early on in the ball python games, when we started seeing more mutations, a big concern of mine became, this is going to get too complicated for people to want to come into this. It's just going to scare them away. Well, I wasn't exactly right about that. It's been pretty cool to see it evolve. I know Kevin McCurley I think one of his books, he tried to lay everything out, by terminology and simplify it, but they were more of the lingo that we had been using, not accurate lingo, I think. So it would be nice if and I don't know how you change anything that we've done for, probably 20 years now. But that would be a challenge, but I think what you're doing is a start. The YouTube videos are a good educational point for us experienced breeders and new people to go check out. So it's just a constant evolving learning process.
Yeah.
So it's just the way it is.
Yeah, I love it.
Yeah, I'm excited about it. You go on with your wrap-up questions, man.
I only have one, Mark. How about you?
One. You usually have like 3 or 4.
How about y'all do me? How about you? How about you come harder than my wrap-up? My wrap-up question is about to kill it right now, so I don't think you're ready.
Okay, go ahead and I'll get ready.
So listen, I always talk about the biggest thing that anyone could do to help them out as far as growth, getting their name out there, you're getting your project out there, is by fucking showing up to shows. Like, you got to go to shows. I think that kind of connects a lot of dots. Where are you at with shows? Like, especially if things are moving in the right direction, and maybe you do build a staff or whatever. Like, do you ever feel like you could ever get to the point where you could be the front face of this at all the big shows and kind of even inform people in person, maybe have some like, some panels or whatnot, to discuss genetics, that sort of thing. I'm just trying to see what the future holds for you and shows.
Yeah, for me, I'm staying extremely busy running samples and developing new tests. But yeah, I see, like, if through this baby season, if things pick up quite a bit and we can hire 1 or 2 more people, then, all of a sudden my ability, my freedom to make it to more shows, that becomes much easier. I certainly would love doing that. I've been going to shows, since, I don't know, 2000, 2002. So I've gone to a lot of shows. I grew up in northern Utah, so I used to go to Salt Lake City show every year. And then,
yeah, but Ben, Dr. Ben, have you been to a show since you've been popping like this? It's a little different when people see your face. Hey man, listen, Mark Bailey has been going to shows for years, but now people know who Mark Bailey is, and they give him mad props whenever he's at shows now. So I think
we have a good time.
It's a good time. It's definitely a good time. And I think this is neat because we want more, and nothing against ball python sales or snake sales, but man, it's cool to go to a show and just gather information that takes you back to your collection versus spending money. And like I said, we all got to spend money on snakes and whatnot, but like I said, I feel like the future holds with with shows. I feel like it's not going to be about snake sales. It's going to be more of marketing, like let's meet and grow from here type of thing. Almost like a convention, like that type of thing. You know what I mean?
Yeah.
I'm saying, laws are, I think, with the future and with animals and how funny it is. I talked about Animal Con. Have you ever heard of Animal Con? By that Brian Barker goes?
Yep.
So last year it was awesome because there was all these vendors there with no animals, but there was just all booths and you're just talking about animals the whole time. You know what I mean? I don't know. I just think it would be a really cool new additive to a show in general, and I can only imagine you guys would be all set up. You could do your thing. Who knows? Fucking from what it seems like, you'd probably be set up next to Canova. I mean, I don't know.
Well, what we did for the last October Tinley, Sean and I just walked around with RGI t-shirts and handed them out to vendors, talked to vendors and handed out t-shirts. So we kind of bounced all around everywhere. And if we were at a booth, it was only for 10 minutes and we were either at the Morph Market booth or the Clutch booth. But we've kind of talked about that, whether we'd want to have a booth or whether we want to just, do what we did, just walk around and talk to people. I don't know. I kind of liked walking around and talking to people, but
it was fun. Yeah.
It's freedom.
It's freedom. But now that more people have seen us, maybe we get stuck, more likely to get stuck, even if we're trying to walk around. I don't know.
Hey, listen, man, you signed up for this, buddy. It is what it is. You know what I mean? You're the face.
I'm happy.
People are loving it, man. All right, Mark, hit him.
Happy.
Not hit him, but hit him with that question.
What's your favorite reptile?
Oh, come on.
That's so hard. That's definitely just like saying what's your favorite thing. It's a lot easier for me if you can boil it down to which one do I think would be the coolest to see in the wild, or which one do I think would be the coolest to
How about
keep.
the wild? How about the wild?
I like that.
So I think if I didn't grow up in the United States, it would have been a
No,
rattlesnake.
it was in the U.S.
Yeah, in the U.S., definitely a rattlesnake.
And
then
what
Gila
particular
monster.
rattle?
A
Which
particular rattlesnake, I'm gonna have to go with the gray-banded rock rattlesnake.
Yeah.
Such a
Cool.
cool animal.
What about
And
you,
I was
Mark?
actually in Arizona for a month studying rattlesnakes, and I saw 55 in 1 month. But I
Whoa.
struck out on Lepidus. I didn't see a single gray-banded rock.
Mark's a
Those
big
are cool. They're pretty secretive, like finding gray-banded king snakes.
But I did
Yeah.
I did get to see like 20 twins.
Oh, twin spotted. Yep.
and he's out. Mark, seriously, is that your final question, buddy? I feel like you do better than that, buddy. Come on,
Oh, you caught me off guard, man.
bro. That was a
Next
long,
time I'll come prepared.
you son of a bitch. Okay, I got to ask you this though. Importation, obviously, I feel like it's needed, maybe not, but what do you feel about animals coming in and out of the country or coming from different parts of the country into the United States? Is that something that we should work on and try to fix, you think? Ben?
You're asking me?
Yeah, you.
That's something I've pretty much always bought captive bred animals, so I've really not thought much at all about that. The part of that I have thought about is how genetic testing could help with potential questions of relatedness or validity or specific species or subspecies, whether it's endangered or not. We could definitely develop tests to help with that.
Yeah.
And I actually have been contacted by, I think it was it manta rays and octopus and some of these other species where they're worried about export and import and developing genetic tests for those. So I've thought about in that area, but I just have not been around imported animals at all. I know almost zero about that.
Well, I think to expand on that, MJ, as far as importing goes, when I first started doing this, we were breeding in captivity to try to prevent some of the importation. And there's I think there's probably a lot less animals being imported now than there ever have been.
Yeah.
The U.S. actually exports quite a few animals. We export a lot of turtles.
Yeah.
Louisiana exports a ton of turtles.
It's crazy. It's like that's the one species of reptiles that people get the most in trouble for, or fucking turtles coming from China, of course, and stuff like that. But it's crazy. You hear obviously it's not out there, but man, I've seen news articles of big names big like importer names that are just getting busted for shit like that. So I never wanted, listen, I've gotten, trust me, a lot of these are imports, but I've never even taken in shipments or I would never want to dabble into any of that. This is too dicey for me. You know what I mean?
Yeah,
and I have no reason adding any imports at this point. Like I'm well beyond that stage. I'm like, like Dr. Ben, like US CBB or bust. Like I want what's born and hatched here. That's it. You know what I mean?
I love
so much
captive
easier.
breeding. I love it with all my heart. Like my wife talked about wanting to get a dog. I was like, well, let's get two and breed them. Like that's just how I am. I'm a breeder. I'm not a keeper or a collector. I'm definitely a breeder. So that's the main reason why I want to buy captive bred. I want to support other people that do the thing that I love.
Yeah, definitely. Well, listen, Mark, I'm gonna let you go. Appreciate you so much. Everyone, please head over to Morph Market because how is your Morph Market looking, Mark? Is it fully stocked?
It's never fully stocked. I'll get to it.
Okay.
Yeah, I've got stuff.
I saw some possets on there that could be tested.
I've got we'll talk. I got a bunch of stuff to send you. So,
but
awesome.
Mark, anyone out there, Mark, who's dying to get your stuff, best way to just be in contact is via Morph Market, you would say, or email, you would say?
Yeah, or text me.
Okay.
You can get my phone number.
All right, cool. All right, man. Well, listen, everyone give it up for Mark Bailey, ladies and gentlemen. Thank you so much, Mark. Have a good night, buddy.
Thanks. Take care, everybody. Nice to visit with you, Dr. Ben.
Yes, you too.
Thanks for the help, man. You killed it, Mark. Thank you.
Thank you. Bye.
All right, buddy. All right, Ben. It's just you and I. You want to know why?
Why?
It's hot seat questions time, buddy. That's what that means.
All right.
Everyone goes through hot seat questions, and you're special, but you're not that special, buddy. You got to get these hot seat questions. And for everyone who's still tapped in, it's not too late to get the likes up or hit the subscribe button. I'm like 2 subs away from 7,000. That'd be great if you hit that subscribe button. But here we go. Hot seat questions for Dr. Ben. Dr. Ben, best way to make you look good with this is answer them as quick as possible. The less explanation, the better. But if you have to explain, you could explain. I'm not saying you can't explain. All right, here we go. Hot seat questions coming in hot. Frozen thought or live?
Live.
Egg cut or no cut? Meaning, would you ever
Yes.
cut? Yes. Okay.
Cut.
On what day for a ball python?
54.
Okay. Red chondro neo or yellow chondro neo?
Red.
Emerald tree boa or green tree python?
Green tree.
Pre first shed meal or post first shed meal?
Post.
Number one or boreal snake?
Green tree. I love green trees.
They're going to say carpet because you've been talking about carpets a lot, and so there's a huge connection with green trees. I see. I love it.
I love green. I've kept 2 or 3, but I never have done really well. Like they've done okay for me, but it's like maybe it's this worry that I shouldn't have, but I'm just worried that I'm not going to do a good job.
Have you met the mayor of Chondrotown yet?
No.
I'll introduce you. His name's Bill Stiegel. He's quite the character.
Oh, yes.
Yes.
I didn't know he had that title, but yes, I do know Bill.
Yeah. The mayor of Chondrotown. Anytime you hear that, that's Bill Stiegel.
He and Sean are good friends. They live pretty close to each other.
Oh, what do you know? See? Exactly. Everyone knows the mayor, man. I'm telling you, he's the man. To spray or mist a ball python or to never spray or mist a ball python?
Yes, in the wintertime because of forced air heating.
Okay.
For me.
Number one substrate for a ball python?
It's old school, but for me, it's aspen.
Aspen, yeah. It's funny because I like the smell of aspen. Some people hate it, but correct me if I'm wrong. With aspen, with good aspen, you avoid all the dust and shit like that. Is that correct? Or why for you is it aspen? Just because it's OG and it's always worked for you? Or
yeah, that's just we didn't have like the coconut, or I didn't know about how to get the coconut options and other things back in the day. Right. I've just always done it. I've done perlite and vermiculite 50-50 mix. That's always worked for me, so I just don't change.
Yay sports or boo sports?
Yay.
Expose animal neglect. Do not expose animal neglect.
Me personally, I want to stay a million miles away from it, but if I'm right in the middle of it, I know what's going on. Expose.
What if you had a podcast and you had nothing to do with genetic like sheds? That wasn't your thing. Your thing was podcasting. Would you do it?
Probably not. Not unless it's someone. If it was someone I thought was going to get away with continuing to do it, unless I speak up, then I would. But as long as I felt like other people are going to take care of it, then it's something that just makes me sad. So I want to not think about it.
Steak or fish?
Steak.
Favorite cut of steak?
Kind of weird, but sirloin.
Why?
I don't know.
Dude,
I had ribeye.
Go ahead.
Yeah, I've had other cuts. I know other people like other stuff. I just like sirloin. I like that's kind of like from that, chewy to not chewy. It's just like what I like.
Hey man, and I love a guy who knows what he likes. You know what I mean? Because so many people are just they don't even know what they like. They just like what others like. But if you stick, stay loyal to yourself. I feel like that's great. Van Halen or Sammy Hagar?
Van Halen.
All right. Favorite genre of music?
I'd have to go alternative.
Nice. Okay, throw one at you here. Green Day or Nirvana?
Nirvana.
Respect. It was all Green Day. It started for me, but Nirvana would just hit different for me. I don't know what it was, man.
That was my very, very first CD was In Utero that I ever bought.
Dude, and you know what's funny? I was too young for that. So my very first, what was the Baby album when he was under the pool? What was the name of the? Smells Like Teen Spirit.
Nevermind.
Nevermind. Yes, Nevermind. But then as I got older, I went back to the very first original, and I was like, "Holy shit, this is what I'm talking about." Yeah. I didn't know Nirvana was getting softer. You know what I mean? I didn't know that they started pretty raw. So, anyways, all right. Little word association. First thing to come to mind: ball python market.
All over the place.
Stuck shed.
Dry.
Reptile investments.
Fun.
Milk.
Good.
Favorite place to vacation?
Beach.
Instagram trolls.
I guess necessary evil.
If you had to choose yes or no, meaning like if it didn't matter if you did this or not, Facebook groups or no Facebook groups?
I like them. I know things get crazy, but I would say yes.
Things definitely get dicey and crazy, man. It all depends what you're there for, right? Like if you want to be involved, well then fuck, good luck. Look what happens. You know what I mean? Hold on. What do you think of Morph Market?
A Morph Market, I don't know what I would do without it. It's definitely, I spent a lot of time on kingsnake.com and on Fauna and stuff back in the day. And as soon as I saw Morph Market, I knew there's no question that was going to be 1,000 times better than what was already there. So yeah, I'm very thankful. I was among the first to get an account and start using it. And I haven't seen anything else coming up that looks better so far.
I mean, it is what it is. Morph Market's it's leading forefront of what we need as far as to get our animals in people's hands, you know?
Yeah, I hope it stays that way.
Yeah, I mean, yeah, me too.
It's definitely been great for me. I happily pay, the yearly amount, and it's always pays for itself, 10 times over.
There's a lot, we were talking about, poking fires, man. There's a lot going on with the new owner of Morph Market. He's off the internet and stuff like that, but Morph Market's still killing it. Like there's still sales. Like I think Morph Market will always be Morph Market no matter who owns it to a certain extent. But, I don't know. Listen, it's what a year it's been. I just got to say, man, but it's only going to get better. I got to tell you, this has been a great episode. Probably the most informative that had to do anything other than breeding. I mean, obviously the snakes, the shed is obviously something related to that, but we didn't, we talked about some really next level revolutionary type stuff for this hobby. And I thought it was an honor to have you on the show. We had over 150 people tapped in at one point. Again, get the likes up. Well, Ben, Dr. Ben, what do you have to say to everyone out there who's rooting for you? Everyone excited to have you here, but more importantly, just the whole reptile industry. What do you have to say to everybody?
Thank you. I'm very thankful. I've had a dream for many years. I've talked to my wife many times about being able to get to a point where I can quit my full-time job and just do reptile stuff. And I was able to do that this last year. And I'm very, very happy, very thankful. I love, even if it is like this last week, I had one night where I was up till 4 a.m. and don't get much sleep. Well, actually a couple nights because of the Desert Ghost episode. I forgot about that, but yeah. Running tests and answering questions, but yeah, I'm very thankful. The questions, the curiosity, all of those things make a big difference. The late nights I have, the people that are excited about it, that's fuel for me.
Yeah, and I personally want to say thank you for putting in those late night works, because it shows that you have to have a passion inside you for something to take you that far. And I got to tell you, man, I have my days where I'm burnt, but if I have something I really need to do that relates to either this podcast or something, I'll fucking do it. I don't care.
Yeah.
And I love my sleep, but something in me keeps me from it just tells me to go. And and I think everyone out there has that in you. Like whatever it is that makes you want to go, you need to do that. And it looks like you found that, and it looks like you've always had it, but thank you so much, Dr. Ben. I know that you're going to be on this show definitely a lot more than just once. I got to tell you, because you're an interesting guy, and I got to say the future of the reptile industry was right here tonight. So thank you to you. Thank you to Sean. I appreciate it. But give it up for Rare Genetics Inc., ladies and gentlemen. It's a wrap.
Thank you.
Dr. Ben, have a good night. Thank you so much. I appreciate it.
Yes, thank you so much. I appreciate it as well.
Later, buddy. I'll talk to you soon.
See ya.
Bye. Whew, what a show, guys. Appreciate it. Everyone who tapped in, quite an audience. Thank you for showing support to, Dr. Ben. This was great. Showing support to Mark Bailey. The entire industry is on fire right now because of this. This is great. So either way, learning's fun. As you guys could tell for tonight's episode, I got a lot of information. This will definitely be something I'm going to go back and watch tonight as I'm downloading it. But for reals, if this was your first time tapping in, if you really enjoyed what you heard on tonight's podcast, hit that subscribe button, man. Smash that subscribe button, hit that notification bell. You'll never miss a show like this. You'll always be on top of it. You'll get alerts. And if you enjoyed this episode as well, you can either drop a super chat. By the way, I love and respect everyone who dropped a super chat. If I didn't get your super chat out there tonight, I'm so sorry. Jamie, I appreciate you so much. Yeah, and you're very welcome. I appreciate it. This is all about kind of bringing information to you guys so we can all grow together as one. Edward, thank you so much for your support as well. I appreciate it. And Morph Valley Reptiles, my dog. I'll ask him this question and I'll get it to you. He's part of the Patreon family, so I'll get that question answered for you. But I appreciate it. But guys, again, thank you so much. Just so you know, I'm doing something pretty awesome this Thursday. Quite a roundtable for the venomous topics. If you're into venomous snakes, or if you want to kind of see like what is the venomous breeder market like, or who's in the venomous breeder community who's like the fucking top dogs. Well, shit. I'll hold on. I'll tell you who that is. I have something really exciting for anyone out there who's interested or anyone tapped in venomous reptiles, either keeping or admiring. But I got to tell you, this is going to be quite a player episode. Okay. All-star venomous lineup. The youngest hitters in this community is coming to the platform Thursday night, 6:00 Pacific Standard Time, episode 356 with Patrick Holmes, Kyle Vargas, and Alexander England. Okay, so set your reminder. Coolest reptile podcast in the world. Again, thank you to everyone who tapped in tonight. Dr. Ben, thank you so much. I appreciate everyone out there. Go follow me on Instagram, TrapTalkGod619, and I'll catch you guys here Thursday night. No, hold on. Hold the phone. Time out. Don't leave yet. I totally lied. And I didn't where is it? Hold on. Where is it? Oh God. You know what, guys? I don't have the thumbnail ready. I feel bad. Guys, anyone who's interested in what's going on with the reticulated python community, tap in tomorrow night. I'm bringing on Lucas. Lucas is the man behind the Slytherin files, the one who put everything together, teamed up with Rodney. So we're still not done tapping in on this. If you really want to see what's going on, set your reminder. Go inside my channel right now. You'll see it. Lab Exotics. It's gonna be nuts. I have to just bring that up. Okay. Anyways, guys, have a good night. Thank you so much. It's a wrap. See you at the top.